Okay, welcome back. Moving through chapter 4. So what I'm going to look at in this video is some organelles which you probably touched on in other classes but not explored functionally in more detail. So those organelles are going to be peroxisomes and vacuoles. So all the other organelles that you hear about in cells, mitochondria, the Golgi, the ER, we'll come back to those a little bit later when we look at some functions in more detail. But just for now, I want to bring the level of your understanding of peroxisomes and vacuoles up to what your current understanding is in respect to mitochondria and those kinds of other more talked about organelles. So let's get going with this. Okay, here's a couple of images of some peroxisomes to start with. Here's a peroxisome on the left shown using electron microscopy. On the right, there is a confocal fluorescence image of peroxisomes, and those are stained in green there. The mitochondria are in red. And one thing to note about the mitochondria, they're not always these nice, discrete oval structures. They can be highly branched in a variety of different colors, and the nuclei there is stained in blue. So that gives you a quick idea of rough sizes, up to a micrometer in diameter, from half a micrometer to a micrometer in diameter, and then you can see their distribution and relative abundance throughout the cell cytoplasm. So let's do peroxisomes first, and then we'll come back to the vacuoles. So peroxisomes have very specialized specific functions. Kind of size-wise, they resemble lysosomes in size and appearance. So if you look at these using electron microscopy, if you look at these using electron microscopy, it's going to be difficult to differentiate a peroxisome from a lysosome. There is something that helps with that, and we'll come to that in a moment. You find peroxisomes abundant in the liver, in cells of the liver, the kidney, brain, lung. So those kinds of tissues you're going to find cells with a lot of peroxisomes. The main function that I want you to focus on is that the peroxisomes take long chain fatty acids. So that is about 22, 24 carbon long fatty acid chains, and they break them down into medium chain fatty acids, which are about 12 carbons, and then those go into the mitochondria to be further metabolized and broken down to produce acetyl-CoA for the Krebs cycle. So that's one of their main functions. They're also involved in detoxifying things like methanol and ethanol. They produce a molecule called plasmalogen, which is involved in the production of myelin, so they're important in nerve cells as well. During a lot of the biochemistry they do, they produce hydrogen peroxide, and hydrogen peroxide is toxic to cells, so peroxisomes have to be able to break the hydrogen peroxide down. So as well as producing it, they break it down using an enzyme called catalase. Now sometimes the catalase will crystallize in the peroxisome, and because it reaches very high concentrations, and so sometimes these peroxisomes have crystalline cores. So that's one way you can discriminate between a peroxisome and a lysosome. Sometimes they have this crystalline core in the middle that you can easily see with electron microscopy. So that's a little bit about peroxisomes. In plant cells, they're well studied in plant cells, particularly in the germination process. So when seeds germinate, as you know, seeds contain high concentrations of fat or high levels of fat, and so those seeds have to break down the fats, and so one of the things they do is they use peroxisomes, and they've got a special name called glyoxysome, and that helps to break down the fat into carbohydrates that can be used then for growth and metabolism. You also find these in leaf tissue as well, and in leaves they're involved in a process called photorespiration. So if you think back to photosynthesis, you talked about, or you learned about an enzyme called rubisco, and you probably really think about what that name means. What that name stands for is ribulose bisphosphate carboxylase oxygenase. And so when carbon dioxide concentrations are high in the environment and plants can take in adequate carbon dioxide, rubisco uses carbon dioxide to add and it adds that carbon from carbon dioxide to RUBP, ribulose bisphosphate, and that's the first step, the fixation step in the Calvin cycle. Now, rubisco isn't necessarily a very good enzyme at what it does, and so if oxygen concentrations are high and carbon dioxide concentrations are low, rubisco can take oxygen, and that's where the oxygenase part of the name comes from. And so it can use oxygen instead of carbon dioxide, and that obviously wastes ribulose bisphosphate and makes the Calvin cycle less efficient. And so peroxisomes are involved in regulating that process. And so here you can see in a plant cell. Here is the peroxisome in the leaf. Plenty of tiny packed chloroplasts, and there's a little mitochondria here, and you can here quite nicely see the crystalline core of catalase. So let's move on now to talk about vacuoles, away from peroxisomes. So a wide range of cells have vacuoles. As a membrane-bound organelle, like the peroxisome and the lysosome that you've learned about before. So these are used for temporary storage or transport. In some cells, they're phagocytic. That means they engulf external material. A internal membrane-bound particle called a phagosome arises, then that type of vacuole, so phagosome is type of vacuole, that phagosome confused with a lysosome, and the lysosome contents are used to break down whatever was in the phagosome. In terms of plant cells, most plant cells, as you know, have a large central vacuole. If you've ever looked at the Elodia in general biology labs, then you will see the large kind of clear central space, and that's a vacuole. It's used for storage, but it also has an important role in maintaining turgor. So making sure the plant stays turgid and so the plant doesn't wilt. So it stores water and ions, and it can also accumulate certain toxins as well, which would otherwise be detrimental or harmful to the plant. And so when your plants wilt, it's because the vacuoles are shrinking because there's not enough water to fill them, and then the cell membrane pulls away from the cell wall, and the process is called plasmolysis, and then the plant cell has inadequate pressure, it loses turgidity, and your plant cell wilts. So there is, on the left, a plant cell with its vacuole. Over on the right here, we have a paramecium. So this little fella's a paramecium, and he's got a couple of different types of vacuoles. So it'll have food vacuoles, and so when the paramecium takes in food through its primitive gullet, food vacuoles are formed, and then they're fused with the lysosomes, and the contents are broken down. But then there are also vacuoles called contractile vacuoles, and they are for regulating water balance. And so water enters a paramecium or an amoeba by diffusion, by osmosis, and water is forced into small vacuoles called contractile vacuoles. And as those vacuoles fill up and swell, they move to the cell membrane, and at a certain point they contract and they expel the water back out of the cell. So this is a mechanism, a homeostatic mechanism for regulating water balance, because obviously these paramecium, they're freshwater critters, they can't get away from the water, and so because the cytoplasm is concentrated, there's an influx of water, and if they didn't expel that water, then they would just burst. So that's a little bit about peroxisomes and a little bit about vacuoles. And when we come back, we will take a look at some viruses, some viroids and prions. And you probably haven't heard about viroids and prions before, so that'll be some new material. I'll speak to you soon.